the topics that we're talking about today, I mean, really could not be more relevant than they are right now. I mean, in the age of LLMs and AI and impending potentially AGI soon enough. I mean, today we'll be talking about the brain and consciousness and informational processing, quantum effects, and really what this all means for us and how to interpret it moving forward. So it's I think extremely relevant. So as for the structure, like Jack said, we'll kind of be out from my end. We'll be sneaking up on the technical bits a bit through kind of grounding it in the hypothesis and how we got to the original hypothesis. And I think that'll help kind of get all of us on board here, moving into the mechanism itself and what this could mean for us and for how to build systems. So it will be some questions at the end. So if you have any thoughts, just write them down and we'll take notes, and hopefully we can get to them at the end. So first of all, I'd love to start off with the shared problems between the brain and LLMs, actually, because I think it's a really interesting place to start, and it kind of gives us a nice thread to work with throughout the rest of the presentation today. But I mean, they face a lot of the similar problems, actually. And first of all, they both have to narrow a vast informational landscape. The brain has to go from all sensory inputs, memories, and its model of the world into something that's coherent and predictive and useful for us. And for LLMs, they have to navigate their entire vector landscapes into something that is then useful for them, which is to provide answers to queries and to be a useful tool for us. And so that's an interesting, aspect up front. But there's also an energy aspect here. So the brain only has about 2.5 watts to work with for informational processing. And I mean, that's less than a light bulb. I'm sure Jack will hit on that later as well, because it's so important. And interestingly enough, while LLMs have, megawatts to work with, each individual query still has a limited token and compute budget. And so it's limited by the forced compression of being as efficient as possible with its answers. So they both have, very limited resources on any given task. And most interestingly, I think, is that both have to interface with an outside world. The brain and, our minds are in our heads. They're in here. And so they have to somehow figure out a way to interface with what's out there. And for us, that seems to be through this ego, through this sense of self and this self-referential processing and this modeling of the world that creates a sense of, self-organization that allows us to maneuver with our external environment. But for LLMs, it's this chat layer, actually. And so it's this reinforcement learning that with human feedback that was teaching the system how to interface with its external environment, which is actually us. And so I think there's some really interesting comparisons there, and probably worth thinking about even more so, because there's a lot to work through. So as for the paper itself, I mean, we can kind of start with where the idea for it originally originated, which was in flow states. And so if you're not familiar with flow, basically just picture a time when everything felt effortless, like sports or music or writing or maybe coding, for example, or gaming. Basically, in these moments of flow, time disappears and effort feels effortless in that sense. So it kind of feels like you can see everything at once, or like 10 moves ahead, or if you're Magnus Carlsen, maybe 1,000 moves ahead. And you're generally performing at the peak of your abilities. And so the question, though, is, well, how do flow states end? What marks this transition? And interestingly enough, what people report is that they often notice how well they're doing. And so you might have a statement like, wow, I can't believe how well this was going, or this is the best I've ever played, or this is an incredible, moment of being efficient, or something like that. And so there seems to be this marking of observation with the ending of a flow state. So the natural question that arose was, well, where else in science is observation correlated with the collapse of a vast informational landscape down to a very, very narrow one? And, well, the obvious answer is quantum mechanics. And so the double slit experiment is kind of like the most famous experiment in quantum mechanics. And basically, if you shoot a photon at a wall with 2 slits without a detector, the photon will behave like a wave and go through both slits, which creates an interference pattern on the wall. And if you can even do this if you shoot 1 photon at a time, it'll distribute as you shoot tons of photons into this pattern. But when you put a detector on the slits, it forces the photon to only go between one of the slits, and it forces it to behave like a particle, not a wave. And so in this environment, the measurement itself, or this, some call it an observation, the measurement or the observation itself is what forces the photon to narrow from all possible paths weighted by action down to essentially one, or being a particle. And so, it sounds a lot like what's happening, what we just described with flow states. So the natural question was, well, could this potentially be a similar or the same physical process? So we have this mode of flow and we have this mode of not flow. So is there precedent for something like this? And in dual process theory, this is already well established. There's two modes of thinking. Daniel Kahneman, Nobel Prize winning economist and cognitive scientist, wrote the book Thinking Fast and Slow, which I would imagine many of you have probably already read. And he describes in it System 1 and System 2. System 1 is this fast, intuitive, parallel mode of thinking, and System 2 is this slower, sequential, linear, logical mode of thinking. So this is really well established in cognitive science, but there's never been a biophysical mechanistic explanation for what these modes actually are or how you switch between them. And so the question basically then becomes, is this just classical fast and slow processing? Basically, are we just turning on more CPUs when we're in System 1 or when we're in flow? Or is there actually something potentially non-algorithmic going on here? And that's where the question, I think, gets a lot more interesting, because the traditional explanations have been that the mind is something akin to a Turing complete type of machine, that any process of understanding or logical reasoning or cognitive function could, in principle, be formalized algorithmically. And that's still very popular today. However, this was famously challenged by Roger Penrose. And so Penrose, who's a Nobel Prize winning physicist, he won the Nobel Prize in Physics in 2020 for his work back a long time ago, for some, with Stephen Hawking, he wrote The Emperor's New Mind in 1989, where he made the argument that the brain can compute non-algorithmically. So I'd love to walk with you through this example to kind of get a good feel for what he was arguing. So basically, if you start with the assumption that the mind computes in a Turing complete manner, then you can assume that everything that Josh can do mathematically, like all of my logical computational processes, could be captured by an algorithm F. So if you assume that's true, then Penrose shows that you can apply Gödel's incompleteness theorem and build a statement G, which is quite simple. It's just this statement cannot be proven by algorithm F. And so Penrose makes it clear that Josh can see why, or understand why statement G must be true, because it's a paradox. But algorithm F, his entire logical apparatus, cannot formalize the understanding. So therefore, Penrose claims that Josh's mathematical understanding can exceed fixed algorithms, and therefore the brain must implement some sort of non-algorithmic process. So, I think it's a really powerful argument, and it still doesn't get enough attention today, in my opinion. But then you have to ask, well, what is this non-algorithmic process? And this is where Roger Penrose postulated, well, it's quantum. And so then the question becomes, well, how? By, what is the biophysics here? To what degree, and what is it doing? So if the brain does indeed have quantum processes, then I think it'd be reasonable to then think that you could somehow model it, at least mathematically, maybe perhaps with quantum statistics, for example. Well, and the field of quantum cognition has done exactly that over the last 25 years. So Busemeyer, Kahneman plus Scotch, for example, have modeled this work incredibly well. And what they found is that under low confidence, quantum models actually predicted behavior more accurately than classical models. However, in high confidence environments, classical Markov models predicted more accurately. So they concluded that neither process alone, quantum or Markov, is sufficient to account for all the conditions, and that both are actually needed for an optimal fit. So they actually asked in their paper, well, how could it switch? And they were speaking from mathematical modeling terms, and they hypothesized that the switch would work through noise in the mental environment. And they hypothesized that this would be where the system would start in a coherent regime and then decohere into a classical regime through interaction with that noise. But they didn't obviously provide a biophysical explanation for what that noise could be, so we'll be exploring that a bit more later. But, then the question becomes, well, if the brain does indeed have both of these modes, like, does that make sense from like a selection perspective? Would it get selected for in that regard? And that's where Vervaeke and Ferraro have done really, really good work from a cognitive science perspective. So they also identified two modes as well. And so we're starting to see a bit of a theme here. They said that evolution and selection pressure would favor organisms that can balance in opponent processing between two computational modes of differentiation, which is kind of like exploration, and integration, which is like organization. And so what they found is that the algorithmic strategies of exhaustive search generally were doomed to failure because of the combinatorially explosive amount of information that the brain has to process. So to put that into context, they used a simple game like chess, where there are 4 times 10 to the 88th paths to checkmate. So famously, Deep Blue, you needed a supercomputer to beat Garry Kasparov, and it took decades to be able to do that, while Kasparov was only using 2.5 watts to compete with megawatts of a supercomputer. And so they're like, well, this doesn't, mathematically make sense for how you could potentially search that much information. So they make the point that the brain, to stay fitted to its environment, has to constantly transcend itself. And they use that language in the paper. I highlighted it there. Transcend itself in its ability to realize what's relevant in the current environment. But this was exactly the point we were just discussing with Roger Penrose, is how can a classical system transcend itself through linear computation? And that's exactly what Penrose was trying to say. It can't. It gets stuck inside algorithm F if you can't transcend it. So we're starting to see a little bit of a pattern here now, that there's these two modes. We have this flow and non-flow, system 1, system 2, non-algorithmic, algorithmic, quantum, classical, differentiation, and integration. And so it's quite likely we have these two modes, quantum and classical, but we still haven't answered, well, what is switching between them and how do they work? And so remember, Busemeyer had proposed that noise in the mental environment could cause this transition. So if Busemeyer is right, well, what would that look like? What is generating the noise? And the next question that emerges here is whether there's evidence in neuroscience, which we haven't examined yet, for any type of transition between these types of modes of thinking. And you can see there at the bottom, there's one we haven't mentioned yet, which is entropy and order from Carhart-Harris, who found this transition at the neuronal level. So they called it the entropic brain hypothesis, that the brain sits at a subcritical point between entropy and order, or high entropy and low entropy. And that they showed how the brain can shift between the modes depending on the environment it's in or on its biophysical conditions. And they showed how the brain sits slightly on the ordered side of things with the ability to shift to the entropic side when needed, which is very similar to what Vervaeke and Ferraro were arguing about, this opponent processing between modes of exploration and more modes of organization. And interestingly enough, they actually identified a part of the brain correlated with this switch. And they said it was the default mode network. So you may be wondering, well, what is the default mode network? Great question. The DMN is the brain's self-monitoring network. And so basically it generates this ongoing internal narrative. It's the felt sense of being a specific person in a specific world and your relationship to the world. So when someone asks you, who are you? It's the DMN that's giving that answer, or the DMN would be highly active while giving that answer, you may say. And so interestingly enough, what they found is that parts of the DMN, including the PCC, operate at 40% above the brain's metabolic baseline. So these are extremely expensive metabolic processes the brain has to use to maintain its sense of self and its organization of the world and its environment. So now the key question is, well, what actually happens when the DMN quiets versus when it's highly active? Is it plausible that the DMN could actually be the switch? And so what they found in their paper was that when alpha decreased in the PCC and in other DMN regions, it was heavily correlated with the dissolution of the ego, of the self, of self-referential processing. And 66% of the variance in ego dissolution was explained by the alpha decreases just in the PCC alone. And so, interestingly enough, also in those moments, the participants reported that their experience took on a supernatural quality. And so, in some sense, they were basically losing this translation layer for the information that was being received by their system that made it predictable. So they were experiencing it as supernatural in some regard as their self was dissolving. So, well, that's nice to know, but what does this actually mean about the switch then? So now we can connect this to flow research. So think back to flow. What were we saying about flow? Well, the flow state collapses when the observation is occurring, when it resumes. So when the self starts evaluating again and thinking, wow, this is an incredible moment. I'm performing the best I've ever played. There's something going on there with the switch. So what's happening in the flow research? Well, that's the picture on the left here, is that Leroy and Cheron in 2020 found that during flow, alpha shifts from the PCC, which is the same region that Carhart-Harris found, to identity, and from identity regions to task relevant regions in about 100 milliseconds. So there's a switch happening here in alpha from identity to task. And this makes it quite clear that at least in some regard, the DMN is involved with some sort of this switching mechanism of sorts. So when it quiets, the metabolically expensive self shuts down, energy relocates, and then this different processing regime emerges. This flow state regime emerges. So at the very least, we can figure out that the DMN is involved at some way with the top end of the switch. So if the DMN is involved, then what would we expect this full mechanism to look like top to bottom? Can we actually start to piece together what we have here and make some predictions? And so what we've established so far is that there's two modes. There's a quantum-like mode and a classical-like mode. And we've established that there's a transition between them involving the noise in this mental environment, and that the DMN is in some way involved at the network or neuronal level in this transition. And this is because the DMN is the most metabolically expensive process the brain maintains, which means that when it quiets, this is a significant amount of energy, per capita or per square meter of the brain that's freed up and now available for other tasks. So based off what we know about quantum mechanics, we can at least make some predictions about what this should look like at smaller scales. So we know from de Broglie's relation that at physiological temperatures, quantum effects become relevant, especially around this 4 nanometer region or so, at nanometer scales. And interestingly enough, we know that tubulin dimers in the cytoskeleton are around that size. And so these structures would be operating right at the scales where quantum behavior would be expected to emerge naturally. So now we know the DMN is clearly involved, and that there would be some sort of top-down and bottom-up pathway that you would expect to connect this DMN activity down all the way to the nanometer scales, potentially all the way down to tubulin dimers, for example. And so these molecular structures would also need to be sensitive to energy supply, considering we saw that so much of this energy per square meter is being relocated when the switch between these modes is occurring. And you would expect them to be affected by electromagnetic noise because that's what the DMN generates. So there would be some sort of connection or causal relation or back and forth in some recursive manner or opponent processing of sorts in that way at that mechanistic level. So, and then realistically, there should be some sort of evidence that connects the substrate down all the way down to the nanometer scales to anesthesia, because anesthesia abolishes consciousness, and this mechanism would inherently be connected to consciousness in some way if there is fundamental quantum processes going on, as Roger Penrose postulated, and as we saw with the fact that Carhart-Harris's work was with psychedelics, and just dissolving the self alone had a transition between these modes. So you would assume that dissolving consciousness would also have something to do with this substrate as well. So
now, considering all of this, we can kind of formulate a formal hypothesis. So I'd love to just kind of read this together here. So now we've basically come to this conclusion that the DMN in some way controls a top-down and bottom-up mechanism that reaches molecular structures at nanometer scales inside neuronal cells down to the tubulin dimers at least. And the electromagnetic noise and the energy dynamics of the DMN in part modulate the conditions for quantum processes at these scales. So whether the system operates in a quantum-enhanced or classical regime would be determined by a recursive loop that propagates downwards from the DMN, but then upwards from those nanometer scales. And this mechanism should be sensitive to disruptions of the energy supply and disruptions by anesthesia. And so on the right side, you can see the mechanism. This is the figure from our paper from Frontiers in Human Neuroscience. And so you can see how far we were able to get and make predictions all the way down to the nanometer scales just from the evidence from cognitive science, from Roger Penrose, from quantum cognition, and then from neuroscience. And you're able to make predictions all the way down to tubulin dimers kind of from the first principles that we're working through off the evidence up top. And so basically that gets us to the mechanism itself. And so to truly understand what's happening at this informational and biophysical level and how the brain can actually do this level of processing at these scales, we've got to go inside the neuron and inside the cell. And that's where Jack comes in. So this basically raises a few questions that will kind of be addressed here throughout the rest of the presentation and maybe into our questions at the end. And for example, is there precedent for this? Do we see quantum energy transfer happening in biological systems at these nanometer scales already? Is it happening in plants, or is it happening in the brain? What are the molecular structures inside of neurons actually doing at those nanometer scales? So how is it actually working in those regards? If the mechanism is connected to consciousness, then what should happen when you disrupt it? For example, with anesthesia. And then we can, begin to discuss how the brain deals with the energy budget and what the energy budget and the constraints look like at these scales. And then how efficient energy transfer would be, why would it be valuable at these scales, and why would it be selected for evolutionarily at these scales in some of the most ancient parts of our body, which would be inside of cells. And so then we can begin to investigate this informational processing capacity and how this would scale up from inside of a cell all the way up to neurons and across the brain. So with that said, I'd love to hand it over to Jack, and he'll enlighten us on that front.
Is it okay? Is it okay to button with a question, or
yeah, maybe you can. Sure, you go ahead. Why don't you ask questions?
Absolutely.
I just want to make sure I understood correctly. Mm-hmm. If I understand what you're saying, when the DMN is more active, then you're saying that on one hand there's more of an organized consciousness, but there's less of quantum activity. That's basically what you're saying.
Yes, exactly.
Okay, so more. Okay. That would be counterintuitive to what I would think about free will, maybe. But okay.
That's quite interesting, because that's where I think the Carhart-Harris's work is so interesting, because they use psilocybin to investigate the DMN. And so what they found is that when you administer psilocybin, the participants, the DMN quiets. It goes essentially flat. And then that's where the alpha oscillations, flatten off. And then that's where they experience the dissolution of self. But this is where they say the mode of exploratory thinking then begins. And this is where they have these magical experiences, or they start connecting ideas they weren't connecting before. So it's a different mode of consciousness in that regard. And so it's a very interesting trade-off here. And that's kind of the idea here. The idea here is that either you can have this very organized consciousness, like what you were saying, that is linear and organized and understandable. So in that sense, language is a linear process. You have to think in grammar, in order, in logic. And so in some ways, it seems that the system is constrained by this linguistic mechanism. And in an alternative mode of thinking, you may think of it as, depending on what your definition of consciousness is, which is a whole different debate. But for example, outside of the self, outside of what Carhart-Harris saw as the ordered consciousness, when the self is dissolved, then this emergent, exploratory, unpredictable mode of processing emerges, in which it's not organized, but it is a conscious experience. And that's where I think it gets very interesting. So hopefully that clarifies that. If there's no other questions, then I guess, Jack, take it away.
Thank you, Josh. That was a great introduction and overview of this area. I think I need permission to share my screen. I'm going to go inside the computing unit, let's say, the elementary aspect of computation, cognition, information processing, namely the neuron. Neuronal cells are known to process information, store memories probably, although we don't know for sure, and they interact incredibly effectively and at a very high rate with other neurons. I just want to mention these 3 books, since we talked about books, that they are kind of setting the stage for many of these topics I will discuss today. Okay, so starting with the human brain, Josh mentioned the 100 billion or so neurons in the brain. There are other cells, of course, almost twice as many other cells, glial cells, astrocytes, and so on. And especially the glial cells are important, which we'll come back to it, namely because of their energy supply, which is quite high by biological standards, although compared to supercomputers is minuscule. But in terms of the human physiology, the brain consumes about 25% of all the metabolic energy, and neurons require a lot of energy for processing. And we can talk about how much and for what, but roughly speaking, we, as mentioned by Josh, about several watts. So 100 watts is the total energy consumption of the human body, 25 for the entire brain, 25% of it, 25 watts, but only about 10% of it or less is used for cognitive processes. The rest is maintenance of temperature, repair, transport of proteins, and so on. Okay, so having said that, each neuron interacts with, on the average, 10,000 other neurons. So it's 10 to the 15th synapses, which leads us to the current paradigms of neurophysiology, which is that everything happens at the synaptic level, and the interactions between neurons via synapses determine the long-term memory, long-term potentiation, and so on. So far, everything is basically classical electrophysiology, just ions flowing in and out, action potentials traveling down axons, and nothing happening inside neurons. And this is where the divergence happens between classical and quantum, in my opinion. All right, so let's go inside. This is an illustration of the neuron itself with the long axon and the soma on the left. Let me see if I can draw. Okay, yeah. So the soma, you have the axon, you have the dendrites and connections to other neurons. In fact, some of them even can connect to 100,000 other neurons. And the density of these interactions determines the brain capacity for all kinds of cognitive functions. And here we have several other things happening. So we have the mitochondria. So energy supply comes from largely from mitochondria. ATP is the quantum of biological energy. They produce ATP molecules, and ATP is, for those who are interested in numbers, about 5 times 10 to the minus 20th joules, which is the quantum of biological energy. You can simplify it to 10 to the minus 20th, which is important because then you can divide by this number and get the number of bits per second you can process. That's the upper limit on the number of processing power. All right, so we have mitochondria, and byproducts of mitochondria are reactive oxygen species, which is important because currently there's a lot of talk about the importance of reactive oxygen species as providing the source of biophotons. I will not have too much time to talk about this, but that you might be interested to look in the literature on the importance of biophotons for potential communication inside the brain, inside neurons, in fact. And then we have here microtubules, single microtubule, and then you have microtubule zoom in into microtubule structure formed from tubulin dimers. And Josh mentioned tubulin dimers. This is about 8 nanometers. Each monomer is about 4 nanometers, so 8 nanometers in length, and that's basically why we think this is a quantum computing unit, because it really is at the limit of quantum collective states. And why is it collective? Because only some of these amino acids forming tubulin are able to be excited in the range of energies that are available, specifically from this and from that, from mitochondria, from reactive oxygen species. We don't know exactly what does it, but at any rate, that's a problem of physics. And that's here you see the action potential. It's a spike that is traveling down the axon, and the current paradigm of neurophysiology tells us that this is where all the action is. And it's an electrical field. It's a field gradient, in fact, which we believe does a lot of other things than just an on-off switch, active, non-active, and that's the current state of affairs. But this electrical gradient interacts with numerous millions of dipoles aligning the surface of the neuron, namely the neuronal membrane. Neuronal membrane is composed of phospholipids, and these phospholipids have relatively large dipole moments, which can be affected by the electric field gradient, and that's kind of a tangent I don't want to go too far into, but it can trigger electromagnetic waves propagating throughout the brain and even interfering and forming these holographic images, which Josh and I have been writing in some other work about. All right. So, but I want to stay here on the topic of microtubules and the potential for being this switch between classical and quantum, because we know a lot about microtubules, and they participate in a lot of different functions in all kinds of cells. All eukaryotic cells have microtubules, but in dividing cells, they form mitotic spindles and they divide the cells. So but in neurons are not non-dividing. So microtubules in neurons are fairly stable and uniquely form parallel bundles. So each microtubule, as I mentioned to you, is formed of tubulin dimers, which would be looked into in more detail in a second. But you'll have in this bundle, you could have tens of microtubules separated by about 90 to 100 nanometers. And the size of a microtubule is about 25 nanometers across and several microns long. And they participate in many things, including transport, exoplasmic transport. So motor proteins move along microtubules inside the axon, and they also interact with ion channels. They can change the activity of ion channels, which we know for sure, but we don't know what role this can play. It may actually change the function of the. So think about the neuron as not a binary switch 0-1, but maybe a quantum computer. And this computer is evolvable. This microtubules may decide is the wrong word, but anyway, I anthropomorphize a bit this, and so they will affect the ability of the membrane to conduct currents and slow down action potentials. We'll come back, and Josh mentioned anesthesia. So anesthetics also interact with microtubules and other things as well. But anyway, here's a lot of work to be done. Okay, so importantly, there's a lot of work, especially done by my collaborator from Los Angeles, UCLA, Nancy Wolf, on remodeling of this neuronal cytoskeleton on learning. When you learn, let's say, a new language or a new skill or a new task, the microtube, not only the neurons interact with other neurons and form stronger synaptic connections and so on, but also inside the neuron, there is a lot of stuff happening, including remodeling of the microtubule architecture. And this is a bit like, reformulating or establishing different connections between your computational units in a microchip, let's say. And so this is known in neurophysiology, okay? And so maybe I'll just skip this because I talked about this is interaction with calcium channels. Calcium are signaling molecules in cells. This is, I should mention the name of Stuart Hameroff, by the way. Penrose was mentioned, and Hameroff is the other half of that duo. Hameroff and Penrose orchestrated objective reduction, but Stuart Hameroff actually started this whole area of microtubule-centered cognition or consciousness. And I want to mention his pioneering work, the book published in 1987 called Ultimate Computing. I read it a few years after it was published, and I couldn't sleep that night because it was so incredibly fascinating. And I recommend even today, with a lot of visionary statements there. And of course, microtubule cytoskeleton is also connected to neuropathologies, pretty much all cognitive diseases, neurodegenerative diseases like Alzheimer's, dementias, bipolar disorders, and so on, are related to some form of malfunction of microtubules in neurons. Okay, so this is indirect. Here you see inside the dendrites where we believe most of the activities take place, including information processing and perhaps memory formation. You see a schematic of microtubules with these little whiskers, which are important. These are so-called C-termini, very short 15 amino acid peptide chains, but highly charged electrically. They carry about 40% of the electrostatic charge of the microtubule, which means that they are highly interactive, and they can be influenced by electromagnetic fields. And this may be one of the means of communicating and even computing signals. Okay, so this is kind of the classical representation of what the microtubule bundle may look like. Basically, RC circuits, very dense, RC circuits, not randomly oriented because this is quite organized, but not perfectly oriented like this diagram. And we've done measurements, actually. I published a lot of papers with my students on the conductive properties and capacitive properties of microtubules. This is beyond speculation. This has been published in serious scientific literature. So they do have the classical component of electricity, conductivity, and charge storage. Okay, so these are some of the papers, and there are many more. And by the way, actin filaments, which are the second sort of most important part of the cytoskeleton, also have interesting conductive properties. They act as diodes, whereas microtubules act as transistors, or can under some circumstances, from the point of electricity alone. But okay, so there's a lot of electrical properties which I have no time to talk about, and even electromagnetic properties. That is how electrical properties change as a result of microtubules being exposed to electromagnetic fields. Let's say in the near infrared, we've done this in ultraviolet, in terahertz, in visible, and all of this matters. And we still don't know, what is the specific mechanism changing these properties, including increased conductivity when you expose them to laser light, and decreased in infrared, et cetera, et cetera. I'll leave it out because there's not much time left. I want to talk about quantum, okay? Because so the classical aspect is RC circuits. You can build microtubules as equivalent RC circuits, let's say. And here you see a microtubule structure with a cross section, which you see here is a hollow interior. This is the 25 nanometer diameter, and you have these antennae, which is C-termini stretched out, but they're very flexible, so they can be movable. They can collapse on the surface, and each of these you can think also as a state, as a physical state, encoding maybe for some information. All right, okay, and we've done again some computational work on this, and let me just accelerate a bit because the time is running out. So that leads to the potential role of microtubules in memory, encoding a lot of information, and some part of information could be in terms of forming interconnections. I didn't talk about this, and Georgie didn't talk about this either, but it's important that these architectures of microtubules, as you can see here from the micrograph of a real cross-section in a neuron, and that's kind of a cartoon version of it. They are interconnected by microtubule-associated proteins called MAPs, and MAP tau is one of them. MAP 2 is another important one. And what happens is when these connections fall apart for various reasons, all kinds of pathologies occur, including Alzheimer's. And tauopathy is one of those. So when MAP tau drops out due to hyperphosphorylation and forms neurofibrillary tangles, microtubules weaken, these bundles weaken, then they disassemble and the neuron stops functioning and memory is lost. So that's the connection to Alzheimer's and therefore connection to microtubules to memory. And okay, so you can do some quick calculations how many memory states you can have in how many bits of memory you can store in microtubules in brain. And I did some rough calculations here. C-termini states, you will have 4 per dimer. Electrons can hop in between dimers, and conformational changes, etc., etc. So this will be per 1 dimer, and there is 100,000 dimers in the neuron, and then there is 100 billion neurons. So you end up with something like on the order of 10 to the 20th bits per brain if you store information in various modes of microtubule states. By the way, this is John von Neumann suggested there's something like 3 times 10 to the 20th, and that was 80 years ago, so maybe 75 years ago. So microtubules would satisfy this kind of prediction made by John von Neumann. Now, this is exaggerated. You cannot have that many transitions between states because you would violate Landauer's inequality. So this is the number of transitions is limited to something like 10 to the 20th or 20th, let's say, bits per second. Bits per second, because of the energy supply. Otherwise, these transitions would cause overheating and the brain would explode. We can't do that. Okay, I'll finish with just give me 2 minutes. I'll finish with the real connection to known states that encode memory. This is an enzyme, calmodulin. So calcium waves trigger the formation of a hollow enzyme called calmodulin kinase 2. It is an enzyme that phosphorylates various states, but includes phosphorylation of microtubules, and this is specific amino acid serine is phosphorylated, and it's shown in turquoise. Okay, and that's the enzyme which we reconstructed with my former student, now Professor Travis Craddock. That's the structure of this enzyme. It looks like a transformer. It walks on microtubules. As it walks, it phosphorylates these amino acids, and by doing so, it encodes memory. This was done by experiments with animals. mice in particular, which were given some inhibitors of CAMK2, and they forgot how to solve mazes and stuff like that. So it's indirect, but it indicates this enzyme in memory formation, and we know that it interacts with microtubules. It looks from above like a snowflake, and it fits beautifully into the microtubule lattice. This is a surface of a microtubule, although it's cylindrical, but you can see a patch of it, and it fits. This is the snowflake again, the calmarginal kinase enzyme, and it's superimposed on the patch of a microtubule lattice, and as it does so, it can phosphorylate up to 6 sites at the same time. And it works like that. It works like a dog, a poodle, or something like that. And as it does, it leaves a trace on the surface. And when you calculate the number of states, it could be up to 1 gigabyte per neuron due to this phosphorylation. Now, okay, so that's information, memory, storage, and this can be actual information of very practical nature, where to connect microtubules with other microtubules through maps, where to change it, and where to send transport, etc. So this could be a practical information. And here you see a kinesin molecule transporting some cargo and moving on a microtubule, maybe being detached, moving to the next one through map, etc. And this was published in 2012, so many years ago now. Okay, I'll finish with, let me just skip quantum computation. So this was speculated, and I guess Josh made a very strong case for quantum computing in the brain in one of the systems. Now, where can it be? And if it's, so that's, of course, hammer of Penrose conjecture, the orchestrated objective reduction, but it was just a conjecture for about 30 years. And now we have some experimental proof that quantum states exist in microtubules, and this is following in the footsteps of quantum biology. Photosynthetic systems like FMO in chlorophyll look very similar to the tryptophan networks in dimers of tubulin and microtubules. This is a tubulin dimer with these 8 tryptophans, which are separated by various distances. And we published now 12 years, is it? Yeah, 12 years ago, a prediction that these could be in quantum states, a collective quantum state. But actually we predicted these to be picosecond timescales, so very fast. But it turned out that with experiments, which you can find in this paper published in 2022 in ACS, we found that, and this will be the last comment I will make, that quantum states which were excited by laser light, 290 nanometer wavelength, in tubulin dimers last about 2 nanoseconds. So actually 1,000 times longer than we predicted, and even longer than in photosynthetic systems. But in microtubules, they last even 3 times more, about 5 to 6 nanoseconds. And through some chemistry I have no time to explain, we also demonstrated the size of the collective quantum state to be, roughly speaking, the size of a dimer, 6 nanometers, which, if you can remember what Josh said, I think he said from de Broglie, it's something like 4 nanometers. We demonstrated 6 nanometers, so it's kind of very close. And now, of course, how you scale up from 6 nanometers to 20 centimeters or so across the brain to have a quantum state, that's the remaining challenge. So the conclusions from this super fast overview of the microtubules and neurons is that microtubules can contain huge amounts of information, can store massive, I would say the entire memory of your lifetime could be stored in the microtubules of neurons in the brain, and they can process information quite fast. And we know now that they support quantum states on the nanoscale, both nanometer and nanosecond time scale. All right, so that's, thank you very much, and that's all I have to say for today.
When we are talking about energy, I was sure for many years until today that the mental energy has nothing to do with like physical joules, calories energy. So do you think, or is there any evidence that this is the same energy that we are talking about? That I'm tired now, that means that I'm burning less ATP molecules than when I'm...
Yeah. Well, I'm a physicist, so according to physics, every bit of information, no matter how encoded, requires a minimum of kT ln 2, a logarithm, natural logarithm of 2 of energy to erase it, or vice versa to encode it. That's in physics. In biology, typically information, and if there's something else, I don't know what it is. If there's something beyond ATP or phosphorylation, it's actually more. It's about 60 kilocalories per mole, which turns out to be 100 kT. So biological energy would be about 2 orders of magnitude greater than physical energy minimum for information encoding. And there's some debate in the literature whether quantum information costs as much energy as classical information, or does it cost at all? And I would say eventually, what Josh was arguing, you eventually have to collapse the state, quantum state, and give an answer which is measurable. And this is physical information indeed. So I would say this estimate of energy consumption for information processing, information encoding is still in place unless somebody comes up with something original that I haven't seen. You have to accept the energy, and yeah.
I was more asking, I'm not talking about like mental energy, like the energy of processing information, but more of like, I'm tired now, I don't have any, like it's the end of the day, I can't process anything else. Is this also associated with actual energy?
Physiological energy?
Yeah.
Well, I can turn it around. I can tell you that maybe this is anecdotal, but I know that grandmasters in chess, they expend a lot of energy when they play chess at this highest level. In fact, they lose weight after playing for hours. So there is this, but yeah, I don't know if there's anything more. Josh, do you know if there's anything more direct about energy demands for mental work?
There's some really interesting research in the flow state side of things. I don't have it in front of me right now, but I would say that there is strong correlation with legitimate like ATP budgets and such, but I don't know if it's been quantified in the way that would be scientific enough to like report in this scenario. But my intuition would assume that it is indeed working with the same energy budget. So the idea here would be is that from, for example, the default mode network, you're limited by this 25 watts. And so if you're, and what we didn't exactly get to the point of talking about fully was that the informational transport, for example, with the anesthesia research, shows that the energy supplies to these lower systems are incredibly important. So you have to maintain an energy pumping at levels to maintain coherent state. So maybe you could talk about that, because that would involve how much energy the DMN is using, and then when that's freed up, it might actually increase the amount of energy available for energy pumping.
Great. Yeah, that's a good point. I think the states that I mentioned, they collapse by themselves. So you have to maintain energy supply to keep them in excited states. And one more thing, during anesthesia, action potentials get weaker and less frequent. So there's a connection in terms of lower energy under anesthesia, and your brain is basically on autopilot. There's no information processing going on. There is no input of more or less no sensory inputs being processed. So that's kind of how I see it.
Thank you.
Regarding the memory, I was always under the impression that there are dedicated regions of the brain for memory, such as the hypothalamus. But if memory is contained in the microtubules, aren't they distributed evenly through all the neurons of the brain?
Well, may or may not be. I think the memory, you may still be right about the hypothalamus being the main area or region of the brain where memory is stored. But that would be simply by signals being directed to that area, and then these enzymes doing the work of encoding inside microtubules of that region of the brain. But I think this is a much larger topic. There's evidence that not just neurons and not just the brain, but there is memory stored in all cells. All cells have some form of memory storage, and there is a work, incredibly interesting work done by Michael Levin from Tufts University in Medford, Massachusetts. He worked with worms, planaria, which regrow their heads. You can decapitate them, and they remember. So, as opposed to these mice treated with calmodulin kinase inhibitors, the worms which get decapitated, they still remember how to solve the mazes and whatnot. I just direct you to the work of Michael Levin, who's done a lot of publishing in this topic.
I could also add something there. Actually, I just recently, a paper got published in Nature by like a ton of, I think it was like tons of authors, like 20 plus authors, that it was titled "A Mosaic of Whole Body Representations on the Human Precentral Gyrus." And they showed that there's the distributed memory of action, or the memory of action is distributed across the entire brain, and that while there's certain areas that specialize in certain actions, you can actually find like picking up a cup is completely delocalized, and so the actions are actually what are localized. Maybe I could link the paper here. I actually have it up. Maybe you'll find that paper valuable, but that is definitely on that topic.
Yeah, so our time is up, and I don't want to abuse your time. So thank you very much for joining us. And I think that the connection between the software and LLMs and biology and physics is just like what makes the world even more interesting than it is now. And we definitely need to explore all those combinations. And of course, neural networks, artificial neural networks, they claim that they're inspired by the actual neurons, but it's like they don't even scratch the surface of.
It doesn't. And please email us if you have any further questions or requests. We'll be happy to respond.
Of course. Thank you very much again.
Yeah,
Bye-bye.
thank you.